So Dr. Hulya Bayir is a professor of pediatrics and chief of the Division of Critical Care and Hospital Medicine in the Department of Pediatrics here at Columbia. Dr. Bayir joined our faculty last year to launch the Children's Health Redox Center at Columbia. Here, Dr. Bayir will continue her transformative research on neuronal injury, oxidative lipidomics, and lipid-based biomarker discovery, which will lead the way to novel redox therapies to protect the brains of critically ill children. Welcome, Dr. Bayir.
So I wanted to start with some history. My history is dating back to 2.8 billion years ago. Oxygen appeared on Earth. And it was because of the production of oxygen by the cyanobacteria and photosynthesis. However, it's toxic. So only the organisms that can actually handle this oxygen then lived on. All the others who could not handle the toxicity of oxygen disappeared. And this is called the Great Oxidative Event. And there is a balance in terms of oxidation and reduction reactions. Redox is the shorthand for oxidation reduction reactions. Oxidation is a key for our survival. We all need oxygen. And when something is oxidized, an electron leaves that molecule, and the molecule that accepts that electron is called reduced. So these reactions, redox reactions, are essential for normal cellular functions. They make energy in the mitochondria. They help us fight infections. They're very important for learning and memory, and they're also important for normal cardiovascular function. However, an increase in the oxidation can cause damage to major molecules in our body, such as our genetic code, DNA, lipids, as well as the proteins. My interest is primarily in lipids, so you'll be hearing a lot about lipids, just fat. So this is the Redox Medicine Research Program in the Redox Health Center that we're establishing. It bridges the fundamental questions that we're asking and we're answering using biochemical and computational tools, experimental models, and clinical studies. A big portion of this program is method development. I'll talk about this method development a little bit more. It's mostly based on mass spectrometry and liquid chromatography. And there are a number of different models, from traumatic brain injury to global ischemia, cerebral ischemia, to acute kidney injury, radiation, acute radiation exposure, as well as NO1 trials that we're interested in. But my talk today will be mostly on traumatic brain injury. Traumatic brain injury is actually the most common, is the major reason for death for children in the United States who sustain a trauma. And trauma is the leading cause of death for children in the United States. Among US children, TBI accounts for 500,000 emergency department visits, 37,000 hospitalizations, and over 2,000 deaths annually. 50% of the children that I see in the intensive care unit with severe traumatic brain injury have dysfunction in neurological function after having severe traumatic brain injury and with the supportive care that we provide them. There is no specific treatment for traumatic brain injury, and the treatment remains supportive. Oxidative damage markers have been associated with poor outcome in adults, but when I first started looking into this, there were no studies in children. And why is this important? Because children are actually more prone to oxidative stress because their enzymes and antioxidants that fight against oxidative stress, such as the superoxide dismutases that get rid of that toxic oxygen derivatives, or glutathione peroxidases, or simply just vitamin C, they don't have enough of it compared to the adults. So they're at more risk for oxidative injury. What we did, we took advantage of a treatment and a diagnostic modality that's done in the intensive care unit. It's called external ventricular drain. Since the skull is a fixed space, when there's a trauma, some kind of contusion, or there's brain swelling. And in order to create more space, the neurosurgeons place an external ventricular drain, which drains the cerebrospinal fluid. It gives us a measurement, intracranial pressure, to guide our treatment, but also gives more space for the brain to complete that swelling process and then go to the repair. We wanted to get the cerebrospinal fluid, which was actually going into the garbage, but look at what's happening because brain is basically bathing in this fluid using comprehensive battery of tools for oxidative stress. What we found was that these squiggly things are a marker for production of free radicals, and it's done using electron paramagnetic resonance spectroscopy. We saw that there's increased production of free radicals. We also saw that there are these molecules such as glutathione, vitamin C, or other antioxidants called antioxidant reserves here. They were declining, and the decline was progressive. And once these antioxidants that combat this oxidative injury, they start declining, there is an accumulation of the damaged proteins, lipids, and DNA. And the biggest damage was in the lipids. And it was early, but also sustained 7 days after traumatic brain injury compared to kids who were control patients who had cerebrospinal fluid taken out to rule out an infection, but didn't end up having an infection. So we established that there is oxidative damage, and this damage is primarily in the lipids. However, this kind of analysis at the time didn't tell us which lipids are being oxidized and how are they oxidized. In order to target these kinds of mechanisms, we need to understand this information because there has been about 40 years of just blanket antioxidant treatments that are in the trials, that are supported by millions of dollars by the NIH, and they did not work. In fact, some of them, even in neonates with vitamin E, cause intracranial hemorrhage. So we really need to understand how is this happening and which lipids are being targeted. So there's an ocean of lipids, and lipids constitute more than half of the human brain in dry weight. So which lipids are the most important targets? We thought that it is the phospholipids because phospholipids are the major building blocks of cells. They form the barriers around the cell. They form the plasma membrane, as well as the membrane around all the small organelles within a cell. They have these two arms, little arms. Those are called SN1 and SN2 position. There's a glycerol backbone and a phosphate group, which a head group that defines what kind of phospholipid this phospholipid is. If there's an inositol here, for example, this is called phosphatidylinositol. If there is a choline, it's called phosphatidylcholine. Ethanolamine, it will be phosphatidylethanolamine, and so forth. Going back to these two arms, they will contain fat that has multiple double bonds. And when fat molecules have multiple double bonds, they're at risk of oxidation. So this then makes sense that we will focus on these phospholipid molecules. And if we consider there are 12 possible chains that can be put in those SN1 and SN2 positions, for the two-legged phospholipids, there will be about 140 molecules. But for a four-legged phospholipid, if all legs have a different color or a different fatty acid, then the number of these molecules will be more than 20,000. So there are many molecules, many lipid molecules. In terms of the oxidation, remember, I told you that when there are double bonds, okay, on this molecule, they will be oxidized. So when there's no double bond or just one double bond, this is an oleic acid, by the way, it's from the olive oil. there will be no oxidation. But when there are many double bonds, the oxidation rate will go up with the number of double bonds. So if this reaction is just random, so there's an oxidant such as hydroxyl radical, everyone heard of it, it will attack indiscriminately all these double bonds and everything will be oxidized. And in addition to all that phospholipids, there are also in the biochemistry in medical school, you learn all these different pathways, the phospholipases that cut an arachidonic acid. Then there's multi-stage oxygenation of this arachidonic acid. And on the right side, there's the free radical mediated oxidation of these phospholipids. So there are even more oxidatively modified species. So how do we study that? In order to study this diverse number of molecules, we had to develop some methods. And we used mass spectrometry because they are very precise balance, joint audio. They weigh molecules and the accuracy. All right. Slides are not advancing.
Not advancing.
The accuracy of this balance goes to the 4th decimal point of this molecule. So we can really measure the weight of these molecules. And here you're looking at a molecule that does those 4 fatty acyl chains with the dog. That's called cardiolipin. And here you're looking at the picture of human brain cardiolipins. On this lower axis, the y-axis, this is the weight of each of these different cardiolipins. And the x-axis, and the y-axis is the amount, how much of that particular cardiolipin is in the brain. So when we did this in the experimental animals, you're looking at cardiolipin again, 2 fatty acyl chains here from left to right, 2 fatty acyl chains from from top to bottom. Cardiolipin in the red, all of them are polyunsaturated fatty acids, so they're prone to oxidation. So this cardiolipin molecule has many polyunsaturated fatty acids, so they are prime target for oxidation. Let's see if other phospholipids also have these polyunsaturated, highly oxidizable polyunsaturated fatty acids, and they do. So all the major phospholipids, as well as cardiolipin, have polyunsaturated fatty acids. So if indeed this is a random phenomenon after traumatic brain injury, all of them will be oxidized. We then developed first a pediatric traumatic brain injury model to look at whether this is the case. On the right side, you're looking at the phospholipid profile. When we see the major phospholipids, phosphatidylethanolamin and choline, they are by far the most abundant phospholipids, and they all have polyunsaturated fatty acids. Cardiolipin, on the other hand, is a minor phospholipid. Let's see what happens after traumatic brain injury. In this immature brain, after traumatic brain injury at an early time point, cardiolipin catches oxidation first, followed by other phospholipids such as phosphatidylserine and phosphatidylethanolamin. So this tells us that this event is non-random, and it starts in the mitochondria because cardiolipin is a mitochondrial phospholipid. We went on and further developed methods to characterize exactly what molecules of this cardiolipin is being oxidized. And we found that there were 166 different kinds of cardiolipins that are being oxidized after traumatic brain injury. This event was not just an experimental model problem, but it also occurred in patients. Here is an example of a contusional tissue that's taken because there was increased intracranial pressure, and just draining CSF, cerebrospinal fluid, was not enough. We had to take, the neurosurgeons took out the contusional tissue, and we saw that there was increased cardiolipin oxidation, whereas the other phospholipids were not oxidized. So what is this cardiolipin? Cardiolipin is first discovered in the 1960s in the heart. It is a phospholipid, but it's very unusual. Most phospholipids will have 2 fatty acyl chains. It has 4 fatty acyl chains. It's kind of has this big body, 2 phosphate groups. So it's 2 phospholipids fused together. And if this plays, you will see that this is a molecular dynamic simulation that because of this configuration, small head, big body, it actually is very important for the mitochondrial crystal structure. If you have ever seen an electron microscopy of mitochondria, it has involutions in the inner mitochondrial membrane. So it's responsible for those involutions. And most of it is primarily in the inner mitochondrial membrane, and it's in the inner leaflet of the inner mitochondrial membrane. The outer membrane has very little of it, and it's another talk that we can discuss why is that so. But what's the mechanism of cardiolipin oxidation? So we discovered over 10 years that it is a molecule, cytochrome C, which is under normal circumstances, it's an electron transporter in the mitochondrial electron transport chain, but it moonlights as a peroxidase when it meets with cardiolipin. And this happens during programmed cell death, specifically apoptosis. These two form a complex, and the substrate is cardiolipin also. So cardiolipin gets oxidized. This complex no longer can be together. Cytochrome C is then released into the cytosol, which is a no return pathway for apoptosis. This leads to well-known apoptosome formation and caspase activation and neuronal death. So this is the full mechanism. So there are, as you see, a number of areas where we can target this mechanism. As I was speaking earlier, if we know, then we can make molecules to target this mechanism. And I'm going to talk about these ones, mitochondria-targeted electron scavengers. These are antioxidants, but specifically targeted to mitochondria. And we chose for this particular group of molecules, we chose nitroxides. Why did we choose nitroxides? Because they prevent formation of superoxide. They accept an electron and they get converted to this hydroxylamine, okay? And then hydroxylamine in turn scavenges radicals and then recycles back to this nitroxide. The reason the peroxidase complex functions is because of formation of hydrogen peroxide. Hydrogen peroxide is needed in that reaction. And hydrogen peroxide is formed from the superoxide. So if we prevent formation of superoxide and formation of hydrogen peroxide, then the peroxidase cycle has nothing to work with. There's no fire. So this is the overall idea. But these molecules can't function unless we send them to the site of action. And the site of action is mitochondria. And we did this in collaboration with Peter Whip, a chemist. We used this gramicidin S. It's an old antibiotic. It forms pores on the membrane, but he cut this antibiotic into half. So now it doesn't form any pores on the membrane, but it brings any molecule that you attach to it. In this case, this is the nitroxide, 4-amino tempo, into the mitochondria. So one thing that's important in neurological drug discovery is blood-brain barrier permeability. So how we tested this is we just gave this 4-amino tempo by itself, or conjugated to our hemigrammicidin intravenously to normal rats. And then we measured the molecule in the cerebrospinal fluid. The measurement details are not important, but as you can see, when you give 4-amino tempo by itself without the grammicidin, hemigrammicidin, it doesn't cross the blood-brain barrier. So in the process, we also discovered how we can overcome the blood-brain barrier permeability. This molecule that I was describing, it's hemigrammicidin nitroxide. It decreased cardiolipin oxidation. Here, as you see, it's XJB because the postdoc who synthesized the molecule, those are the first letters of his name, XJB. So it decreased cardiolipin oxidation, which is the biochemical target. It also decreased improved histological and functional outcome. It's not enough in neurological diseases that we improve some histological outcome, but we also have to show that there's actually an effect on the function. Otherwise, it's not going to be helpful. In this slide, you're looking at cognitive performance assessment in animals. These are 24-day-old animals after pediatric traumatic brain injury. And you can see on this blue line, this is learning over time to find a hidden platform in this tank with cues that are on the wall. And the animals that had trauma and just fecal, they didn't learn as fast as the animals who didn't have trauma, just anesthesia, and the animals who got the XJB. So we have some of these homologs that are not good, but some of them are good. So we're working on them to improve them. In the last 3 minutes, I'm going to talk about, I showed you that we grind up the tissue and we look at all these signals. But that kind of approach doesn't tell you where these things are happening in the brain or in a cell. So in order to do this, we develop methods to image lipids using no tags. There are no antibodies for lipids. You can't do this any other way, but using imaging mass spectrometry techniques. These are not in vivo. These are in tissue slices, brain tissue slices that are frozen tissue, coated with something called matrix that helps the molecules to ionize during this process. You then shine light, basically laser. The molecules get excited, they ionize, and they're taken up by the mass spectrometry. Each line, now you remember these lines, now you can draw a picture of each line on this spectrum, and that's called imaging mass spectrometry. Okay. Too bad the video isn't playing. They will move on. I was going to show you how these images are generated. Now we're stuck. We can't move forward or. Okay. Okay, so you can see that the area of impact is here. This looks at cardiolipin loss. It's not just in the area of impact, which is the cortical area, but it's also in the hippocampus, and it's in the midbrain as well. And it's primarily the polyunsaturated fatty acids. I'm not going to talk about this super duper other strategy that we can do. You can ask me questions about it. This is called secondary ion mass spectrometry. With that previous technique, we can only look at 50 micron down to 20 micron. With this technique, we can go down to 1 micron, so we can actually look at cells. So, and also we can look at proteins and metabolites, so it's multi-omics. So we learned that redox reactions have important signaling roles. Lipid peroxidation is non-random and enzymatically catalyzed, and targeting these reactions might be beneficial. We also have a lot of studies on other type of cell death, in addition to apoptosis. These are the 3 posters that my lab members, Dr. Samovich, Dr. Korkmaz, and Dr. Demir are presenting. So please visit their poster. Thank you.